Cell, the unit of life, cell theory and prokaryotic cell. All organisms are composed of cells. All unicellular organisms are composed of a single cell, while others are composed of many cells and are called multicellular organisms. What is a cell? Unicellular organisms are capable of existing independently and perform the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, in all living organisms, cell is the fundamental structural and functional unit. A live cell was first seen and described by Anton von Leeuwenhoek. The nucleus was later discovered by Robert Brown. All the structural details of the cell was revealed by the invention of the microscope and later the electron microscope. Cell theory. A large number of plants were examined in 1838 by Matthias Schleiden, a German botanist. He observed that all plants are composed of different kinds of cells which form the tissues of the plant. Later, a British zoologist, Theodore Schwann, studied different types of animal cells and he reported that cells had a thin outer layer which is today known as the plasma membrane. Based on his studies on plant tissues, he also concluded that a unique character of the plant cells is the presence of cell wall. On the basis of this, a hypothesis that the bodies of animals and plants are composed of cells and products of cells was proposed by Schwann. Together, the cell theory was formulated by Schleiden and Schwann. This theory, however, did not explain the formation of new cells. However, the cells divided and new cells were formed from pre-existing cells was first explained by Rudolf Virchow. The hypothesis of Schleiden and Schwann was modified and he gave a final shape to the cell theory. The cell theory is understood today as all living organisms are composed of cells and products of cells. All cells arise from pre-existing cells. An overview of cell. Earlier, you have observed under the microscope the cells in an onion peel or human cheek cells. Let us recollect this structure. A typical plant cell, the cell of an onion, at its outer boundary has a distinct cell wall and just within it has the cell membrane. In human cheek cell, the delimiting structure of the cell is an outer membrane. Inside each cell is a nucleus, which is a dense membrane-bound structure. The nucleus contains the genetic material DNA, which is present in the chromosomes. The eukaryotic cells have membrane-bound nuclei, whereas prokaryotic cells lack a membrane-bound nucleus. A semi-fluid matrix called cytoplasm occupies the volume of the cell in both prokaryotic and eukaryotic cells. The main arena of cellular activities in both the plant and animal cells is the cytoplasm. To keep the cell in the living state, various chemical reactions occur in it. The eukaryotic cells besides the nucleus have distinct other membrane-bound structures called organelles like the endoplasmic reticulum, the lysosomes, Golgi complex, microbodies, mitochondria and vacuoles. Such membrane-bound organelles are absent in the prokaryotic cells. In both prokaryotic as well as eukaryotic cells, ribosomes are present which are non-membrane bound organelles. Ribosomes in the cell are not only found in the cytoplasm but also within the two organelles chloroplasts and mitochondria and on rough endoplasmic reticulum. Another non-membrane bound organelle called centriole is present in the animal cell which helps in cell division. The cells differ in size, shape, and activities. For example, smallest cells, the mycoplasmas, are only 0.3 μm in length, whereas bacteria could be 3 to 5 μm. The egg of an ostrich is the largest isolated single cell. Human red blood cells among multicellular organisms are about 7.0 μm in diameter. Some of the longest cells are the nerve cells. In their shape, cells also vary greatly and they may be polygonal, disc-like, cuboid, columnar, thread-like, or even irregular. The cell may vary in shape with the function they perform. Prokaryotic cells. The prokaryotic cells are represented by mycoplasma, the PPLO or pleuropneumonia-like organisms, bacteria, blue-green algae. These organisms are generally smaller and compared to the eukaryotic cells, they multiply more rapidly. They greatly vary in shape and size. The bacteria are of four basic shapes, namely Bacillus, Coccus, Vibrio, and Spirillum. Even though prokaryotes exhibit a wide variety of shapes and functions, the organization of the prokaryotic cell is fundamentally similar. In all prokaryotes, the cell membrane is surrounded by the cell wall. The cytoplasm is a fluid matrix filling the cell. No well-defined nucleus is present. The genetic material is not enveloped by a nuclear membrane and is basically naked. Many bacteria, in addition to the genomic DNA, have a small circular DNA outside the genomic DNA, and the smaller DNA are called plasmids. To such bacteria, the plasmid DNA confers certain unique phenotypic characters, and resistance to antibiotics is one such character. In eukaryotes, nuclear membrane is present. In prokaryotic cells, except for ribosomes, no organelles like the ones in eukaryotes are found. In the form of inclusions, prokaryotes have something unique. Mesosome, which is a specialized differentiated form of cell membrane, is characteristic of prokaryotes, and they are essentially infoldings of cell membrane. Cell envelope and its modifications. A chemically complex cell envelope is present in most prokaryotic cells, particularly the bacterial cells. A tightly bound three-layered cell envelope consists of the outermost glycocalyx followed by the cell wall and then the plasma membrane. A distinct function is performed by each layer of the envelope and they act together as a single protective unit. On the basis of the differences in the cell envelopes and the manner in which they respond to the staining procedure developed by Gram, the bacteria can be classified into two groups, namely those that take up the Gram stain are Gram positive and the others that do not take up the Gram stain are called Gram negative bacteria. Among different bacteria, glycocalyx differs in composition and thickness. It could be the slime layer in some, which is a loose sheath, while in others it may be a capsule, which is a thick and tough. The shape of the cell is determined by the cell wall, and to prevent the bacterium from bursting or collapsing, the cell wall provides a strong structural support. The semi-permeable membrane is a plasma membrane and it interacts with the outside world. The plasma membrane is structurally similar to that of the eukaryotes. The mesosome, a special membranous structure, is formed into the cell by the extension of plasma membrane. The vesicles, tubules, and lamellae are the extension of plasma membrane. They help in the formation of cell wall, replication of DNA, and distribution to daughter cells. They also help in secretion processes, respiration, enzymatic content, and to increase the surface area of the plasma membrane. In some prokaryotes like cyanobacteria, there are chromatophores, the other membranous extensions into the cytoplasm which contain pigments. The cells of the bacteria may be motile or non-motile. They have thin filamentous extensions from their cell wall called flagella if they are motile. In the number and arrangement of flagella, the bacteria show a range. The flagellum of the bacteria is made up of three parts: filament, hook, and basal body. The longest portion forms the filament and it extends from the cell surface to the outside. Besides flagella, pili and fimbriae do not play a role in motility, but they form surface structures of the bacteria. The pili are made of a special protein and they are elongated tubular structures. The fimbriae sprout out of the cell and are small bristle-like fibers. In some bacteria, they help to attach the bacteria to the host tissues and also to the rocks and in streams. Ribosomes and inclusion bodies associated with the plasma membrane of the cell in prokaryotes are the ribosomes. The prokaryotes are about 15 nm by 20 nm in size and they are made of 50S and 30S subunits which put together form 70S prokaryotic ribosomes. The site of protein synthesis are the ribosomes. To a single mRNA, several ribosomes may attach and form a chain called polyribosomes or polysomes. A polysome contains the ribosomes and the ribosomes translate the mRNA into proteins. Inclusion bodies. In prokaryotic cells, reserve material are stored in the form of inclusion bodies in the cytoplasm. These lie free in the cytoplasm and are not bounded by any membrane system. Example: cyanophycean granules, phosphate granules, and glycogen granules. In blue, green, purple, and green photosynthetic bacteria, gas vacuoles are found. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content.